I study rare disease and elite athletes, which are both on the spectrum in terms of both human performance and rare disease. So I study the genetic aspect and the genetic component of these unique individuals, and I try to understand how to improve the performance through different means or to cure people with very rare genetic disorder. Our current project is actually to provide a means to screen probiotics and their effect on muscle health, muscle performance, and to determine what will be the best formula to apply to improve muscle performance and health in general. The application of our project goes, of course, with rare individuals that have disorder like metabolic rhabdomyolysis, but also elite athletes or athletes in general, which try to improve their performance. We have developed cellular models of the gut barriers and also a cellular model of muscle. So we have developed a mini muscle in vitro, which replicates the different function of muscle like contractility and so on, and they are really more potent than the current models that we are developing in the field. If you look at the gut barrier, we have included goblet cells that reproduce mucus, and this mucus is very important for the gut microbiota. So far this was a limitation in the different models that were presented. And in terms of the mini muscle, we call this like really mini muscle because it's 8 millimeter muscle that we are able to build in vitro. This mini muscle will be used to test the effect of different strains, in particular to determine if they promote the contractility, they prevent fatigue, and other parameters that are crucial for muscle health. The motivation to go on this area of investigation is first we start to screen a lot of different bacteria, and there's potentially some bacteria that will have an effect, but we need to know which one. And if you think about the gut microbiota as a whole, there's billions of different bacteria. So we have to investigate them one by one, but also as a combo in order to determine novel formulation. This work cannot be done in vivo and can only be developed through in vitro studies, which have the power to screen large amount of bacteria and different formulation. So the collaboration with Lallemand was particularly interesting because we started to speak about the model, we started to speak about how to use them, and we entered into a co-creation. And this project was not just a proposition that we did to Lallemand, it was a co-creation between us and the research group of Lallemand. It has shown a lot of power because we definitely can address some key answers of the company. The collaboration is particularly interesting because everybody brings his own strength. We bring the model, Lallemand brings the problem to solve, and together we're here to fix the problem and bring some solution, especially regarding health for different individuals that are developing disease or that are healthy too. I think the finding of our research have a huge potential because we can determine some new formulation. Our model also are developed from human skin fibroblasts, so we can personalize the approach and integrate the full spectrum of the genetic of each individual into the picture. And in that regard, this collaboration is probably unique because we will approach the complementation and the supplementation from an individual point of view. Using our model and testing the effect of the gut microbiota metabolites, we have shown that there is some epigenetic effects that could translate into long-term effect on well-being and muscle longevity, which will be an asset at the end of the day for people and a proper aging. The interest of working on athletes is the democratization of research and the applicability to a wealthy population that will be looking for well-being and a healthy aging, in particular in preventing sarcopenia or other disorder affecting muscle mass and function later in life.